Hello everyone, I am Mohammed Hamama, and this is your ASCP preparation camp. In this camp, we will go through each topic on the ASCP lecture list. In today's video, we will be discussing intrinsic defects leading to increased erythrocyte destruction. Intrinsic defects leading to increased erythrocyte destruction. A significant number of disorders fall under the category of intrinsic hemolytic anemias, where defects within the red blood cells lead to early destruction and anemia. Such intrinsic disorders can be classified as red blood cell membrane abnormalities or red blood cell enzymopathies, and the majority of them are genetically inherited. Before we start, we will recall some pieces of information about red blood cell membrane structure and function. The red blood cell, RBC, maintains a biconcave discoid shape that is important for its normal function and survival in peripheral circulation for 120 days. The plasma membrane, which is a lipid bilayer embedded with proteins, is crucial in maintaining the RBC's shape. The membrane serves as a barrier to separate the RBC's interior from the blood plasma and is regulated by proteins that serve as pumps and channels for the movement of ions and other materials. The membrane also has various proteins that act as receptors, RBC antigens, enzymes, and support for the surface carbohydrates to form a protective glycocalyx. Transmembrane proteins and an underlying cytoskeleton provide structural integrity, cohesion, and mechanical stability to the RBC. RBCs must have deformability to maneuver through narrow capillaries and splenic sieves without premature lysis. Deformability is enabled by the RBC's geometry, the elasticity of its membrane, and its cytoplasmic viscosity. Two transmembrane protein complexes, the ankyrin complex and protein 4.1 complex, provide vertical structural integrity to the cell by anchoring the lipid bilayer to the underlying spectrin cytoskeleton. The cytoskeletal proteins alpha spectrin and beta spectrin interact laterally with each other to form antiparallel heterodimers that link with other spectrin heterodimers to form tetramers, which provide horizontal mechanical stability. Factors that impact the elasticity of the cell are not entirely clear, but interactions between the spectrin dimers and their junctional complexes allow for movement as the RBCs stretch and bend. The cytoplasmic viscosity depends on the concentration of hemoglobin, as well as the maintenance of the proper cell volume by the normal functioning of various channels and pumps. A defect in the RBCs that changes the membrane geometry, its elasticity, or the viscosity of the cytoplasm affects RBC deformability and can result in premature hemolysis and anemia. Hereditary red blood cell membrane abnormalities. Most defects in the red blood cell membrane that can cause hemolytic anemia are hereditary, but acquired defects also exist. Hereditary RBC membrane defects are historically classified by morphologic features, including hereditary spherocytosis, characterized by spherocytes. Hereditary elliptocytosis, characterized by elliptical RBCs. Hereditary pyropoikilocytosis, a variant of hereditary elliptocytosis, characterized by marked poikilocytosis and heat sensitivity. Other less common membrane disorders include hereditary ovulocytosis, overhydrated hereditary stomatocytosis, and dehydrated hereditary stomatocytosis, also called hereditary xerocytosis. Hereditary membrane defects can be classified as those that affect membrane structure, and alter geometry and elasticity, and those that affect membrane transport, and alter cytoplasmic viscosity. The membrane structural defects can be further divided into those that affect vertical membrane protein interactions and those that affect horizontal membrane protein interactions. Mutations that alter membrane structure. Hereditary spherocytosis. Hereditary spherocytosis is a type of hemolytic anemia caused by defects in proteins that disrupt the vertical interactions between transmembrane proteins and the underlying protein cytoskeleton. Hereditary spherocytosis has two modes of inheritance. 75% of cases are inherited as an autosomal dominant trait and 25% of cases are inherited in a non-dominant manner. The defective proteins associated with hereditary spherocytosis include ankyrin, band 3, alpha-spectrin, beta-spectrin, and protein 4.2. Hereditary spherocytosis is characterized by loss of membrane and decreased surface area to volume ratio, resulting in spherocytes and polychromasia. The severity of hereditary spherocytosis can vary from asymptomatic to severe, but typical features include splenomegaly, jaundice, and anemia. Pathophysiology. Hereditary spherocytosis is caused by gene mutations that result in defective proteins disrupting the vertical linkages between the lipid bilayer and the cytoskeletal network. Hereditary spherocytosis phenotype can result from various mutations in 5 known genes. A primary mutation in one gene may have a secondary effect on another protein in the membrane due to the vertical interactions of the transmembrane proteins and cytoskeletal protein lattice. RBCs lose unsupported lipid membranes over time because of local disconnections of the lipid bilayer and underlying cytoskeleton, forming vesicles that are released with little loss of cell volume. RBCs acquire a decreased surface area to volume ratio, become spherical, and lose deformability, resulting in decreased survival in the spleen. RBC membranes in hereditary spherocytosis have abnormal permeability to cations, particularly sodium and potassium, which is likely due to disruption of the integrity of the protein cytoskeleton. The cells become dehydrated, increasing viscosity in cellular dehydration, regardless of the type of primary mutation causing the hereditary spherocytosis. Clinical and laboratory findings. Symptomatic hereditary spherocytosis has 3 main clinical manifestations: anemia, jaundice, and splenomegaly. Symptoms can appear at any age, and there is variation in their severity. Silent carriers of hereditary spherocytosis have no symptoms and normal laboratory findings, usually identified only if they are parents of a child with hereditary spherocytosis. About 20 to 30% of hereditary spherocytosis patients have mild hereditary spherocytosis, with normal hemoglobin levels but subtle signs of hereditary spherocytosis such as slightly elevated bilirubin levels and increased respiratory counts. Approximately 60% of patients have moderate hereditary spherocytosis with incompletely compensated hemolytic anemia, hemoglobin levels greater than 8 grams per deciliter, serum bilirubin over 2 mg per deciliter, respiratory counts in the range of 6% to 10%, and spherocytes on the peripheral blood film. About 5% to 10% of patients have moderate to severe hereditary spherocytosis with hemoglobin levels usually in the range of 6 to 8 grams per deciliter, serum bilirubin between 2 to 3 mg per deciliter, and reticulocytes counts greater than 10%. About 3% to 5% of patients have severe hereditary spherocytosis, with hemoglobin levels below 6 grams per deciliter, serum bilirubin over 3 mg per deciliter, and reticulocytes counts greater than 10%. Patients with severe hereditary spherocytosis usually require regular transfusions. Hereditary spherocytosis is diagnosed by the presence of spherocytes on the peripheral blood film, along with other RBC morphologic variants that may be observed in some types of mutations. Hereditary spherocytosis patients have an increase in MCHC and RDW and may have hyperchromic or hyperdense RBCs. Biochemical evidence of extravascular hemolysis may be present in moderate to severe forms of hereditary spherocytosis, and the extent is dependent on the severity of the hemolysis. Bone marrow analysis is not required for hereditary spherocytosis diagnosis but shows hyperplasia due to the increased demand for RBCs to replace the circulating spherocytes that are prematurely destroyed. Laboratory tests. To diagnose hereditary spherocytosis in patients with a family history of the disease, splenomegaly, an increased MCHC and reticulocytes count, and spherocytes on the peripheral blood film, no additional testing is required. However, in cases where hereditary spherocytosis is suspected but the family history and mode of inheritance are not clear or there are atypical clinical and laboratory findings, special testing is necessary to confirm the diagnosis. It is important to note that no single test can detect all cases of HS, and a combination of tests is necessary for a definitive diagnosis. Osmotic fragility test. The osmotic fragility test shows how easily red blood cells break apart in blood samples with lower surface area to volume ratios. The test involves adding blood to tubes with increasingly hypotonic sodium chloride solutions. Water enters and leaves the RBCs until equilibrium is achieved. In 0.85% NaCl, water enters and leaves the cell equally because the intracellular and extracellular osmolarity is the same. In hypotonic solutions, more water enters the cell to dilute the intracellular contents until equilibrium is reached, causing the cells to swell. RBCs lyse in increasingly hypotonic solutions. Spherocytes, which have a decreased surface area to volume ratio, lyse in less hypotonic solutions than normal-shaped RBCs, leading to increased osmotic fragility. The procedure involves mixing a standard volume of fresh, heparinized blood with NaCl solutions ranging from 0.85% to 0.0% in increments of 0.05% to 0.1%. After a 30-minute incubation at room temperature, the tubes are centrifuged, and the absorbance of the supernatant is measured spectrophotometrically at 540 nanometers. The percent hemolysis for each tube in the osmotic fragility test is calculated using this formula. AX% is the absorbance in the tube being measured, a 0.85% is the absorbance in the 0.85% NaCl tube, representing 0% hemolysis, and a 0.0% is the absorbance in the 0% NaCl tube, representing 100% hemolysis. The percent hemolysis for each percent NaCl concentration is plotted, and an osmotic fragility curve is drawn. Normal biconcave RBCs show initial hemolysis at 0.45% NaCl, and complete hemolysis generally occurs between 0.35% and 0.3% NaCl. If the curve is shifted to the left, the patient's RBCs have increased osmotic fragility, and initial hemolysis begins at a NaCl concentration greater than 0.5%. Conversely, if the curve is shifted to the right, the RBCs have decreased osmotic fragility. Decreased osmotic fragility is found in conditions with numerous target cells, such as thalassemia. The presence of a tail on the osmotic fragility curve reflects a subpopulation of the most fragile cells, which are most conditioned by the spleen. After splenectomy, the osmotic fragility improves and the subpopulation of conditioned cells disappears. The incubated osmotic fragility test, which involves incubating blood at 37 degrees Celsius for 24 hours before the test, allows for the detection of mild cases of hereditary spherocytosis. The test is time-consuming and requires a fresh heparinized blood specimen, and accurately made NaCl solutions. The test has a lack of sensitivity, with a modest increase in sensitivity with the incubated test, and is also nonspecific, unable to differentiate between hereditary spherocytosis and spherocytosis caused by other conditions. These disadvantages have led some not to recommend this test for routine use. The eosin 59 maleimide EMA. The eosin 59 maleimide binding test is a proposed alternative for confirming hereditary spherocytosis. EMA is a fluorescent dye that binds to specific transmembrane proteins in the RBC membrane. The test is performed using flow cytometry and measures the mean fluorescence intensity MFI. HS patients show a lower MFI compared to normal controls in patients with spherocytes due to immune-mediated hemolysis. The test has a sensitivity and specificity of 93 to 96% and 94 to 99%, respectively. Positive tests may occur in rare conditions such as congenital dyserythropoietic anemia type 2, Southeast Asian ovulocytosis, and hereditary pyropoikilocytosis. The test is suitable for low-volume pediatric specimens, can be performed within 3 hours, and specimens are acceptable for analysis up to 7 days after collection. Gating can be used to eliminate interference from transfused or fragmented RBCs. Laboratories disagree on the %MFI decrease cutoff value for hereditary spherocytosis, and standardization is needed. Additional tests. Additional tests may be necessary in cases of atypical hereditary spherocytosis to identify defective proteins. Sodium dodecyl sulfate polyacrylamide gel electrophoresis can be used to identify membrane protein deficiencies by separating various proteins in solubilized RBC membranes with quantitation of proteins by densitometry. Radioimmunoassay can be used to quantify membrane proteins. Osmotic gradient ectocytometry can determine variation in membrane surface area and cell water content by recording the laser diffraction pattern of a suspension of RBCs exposed to constant shear stress in solutions of varying osmolality. Acidified glycerol lysis test measures the amount of hemolysis after patient RBCs are incubated with a buffered glycerol solution at an acid pH, but it lacks specificity. In the autohemolysis test, patients' RBCs and serum are incubated with and without glucose. HS hemolysis is 10% to 50%, which corrects but not to the reference interval when glucose is added. The hypertonic cryohemolysis test calculates percent hemolysis after the patient's RBCs are incubated in buffered 0.7 moles per liter sucrose, first at 37 degrees Celsius for 10 minutes and then at 0 degrees Celsius for 10 minutes. Hereditary spherocytosis RBCs have greater than 20% hemolysis. Molecular techniques for detection of genetic mutations are not usually required, but polymerase chain reaction and nucleic acid sequencing can be performed to identify mutations. Please don't forget to subscribe to our channel, activate notifications to get our new videos. If you like our content, please press the like button, and share the video with your friends. If you have any questions, leave a comment below. Hereditary elliptocytosis. Hereditary elliptocytosis is a group of hemolytic anemias caused by defects in proteins that disrupt the horizontal or lateral interactions in the protein cytoskeleton. The disease is mostly inherited in an autosomal dominant pattern, but there are also a small number of autosomal recessive cases. The proteins that are most commonly affected in hereditary elliptocytosis are alpha-spectrin, beta-spectrin, and protein 4.1. The loss of mechanical stability of the membrane leads to the characteristic elliptical shape of the red blood cells, which range from few to 100% elliptocytes. In severe cases, schistocytes may also be present. About 90% of individuals with hereditary elliptocytosis are asymptomatic, while the remaining 10% experience moderate to severe anemia. Hereditary elliptocytosis reportedly exists in all its forms in 1 in 2,000 to 1 in 400 individuals, but the majority of cases are asymptomatic, and the actual prevalence is not known. Hereditary elliptocytosis is more common in Africa and Mediterranean regions, where there is a high prevalence of malaria. The prevalence of certain spectrin mutations associated with hereditary elliptocytosis in West Africa is between 0.6% and 1.6%. The molecular basis for the association of spherocytosis and malaria is unknown. The inheritance pattern in hereditary elliptocytosis is mainly autosomal dominant, with a small number of autosomal recessive cases. Pathophysiology. Hereditary elliptocytosis is caused by gene mutations that disrupt the horizontal linkages in the protein cytoskeleton and weaken the mechanical stability of the RBC membrane. Hereditary elliptocytosis can result from mutations in at least 3 genes. The mutations in SPTA1 and SPTB disrupt spectrin dimer interactions, while mutations in EPB41 weaken spectronactin protein 4.1 junctional complexes. The extent of the disruption of the spectrin dimer interactions is associated with the severity of clinical manifestations. RBCs in hereditary elliptocytosis are biconcave and discoid at first, but become elliptical over time after repeated exposure to shear stresses in the peripheral circulation. Severe disruption of the protein cytoskeleton can cause cell fragmentation, membrane loss, and a decrease in surface area to volume ratio, reducing RBC deformability. Damaged RBCs become trapped or acquire further damage in the spleen, resulting in extravascular hemolysis and anemia. Heterozygous patients are asymptomatic with normal RBC lifespan, while homozygous or compound heterozygous patients can have moderate to severe anemia that can be life-threatening. A mutation database for hereditary elliptocytosis lists 46 different mutations. Hereditary elliptocytosis patients have defective proteins that disrupt the membrane's mechanical stability, causing RBCs to become elliptical. Mutations in 3 genes, SPTA1, SPTB, and EPB41, are responsible for hereditary elliptocytosis, with SPTA1 mutations being the most common, 65% of cases. RBCs in He patients have reduced thermal stability and can become fragmented upon heating. Hereditary pyropoikilocytosis (HPP) is a severe form of hereditary elliptocytosis and is either homozygous or compound heterozygous. The Leach phenotype is caused by a mutation in the genes for glycoprotein C, GPC, resulting in a lack of GPC in the RBC membrane. The absence of GPC causes Gerbich antigens to be absent as well. Heterozygotes have normal RBC morphology, and homozygotes have mild elliptocytosis but no anemia. The elliptocyte morphology in homozygotes may be due to a defect in the interaction between GPC and protein 4.1 in the junctional complex. Clinical and laboratory findings. Most patients with hereditary elliptocytosis show no symptoms, while about 10% experience moderate to severe anemia. Some patients may have mild hemolytic anemia, while others may develop transient hemolysis due to other conditions. Asymptomatic patients may be diagnosed after a peripheral blood film examination. Heterozygous parents with undiagnosed asymptomatic hereditary elliptocytosis may have offspring with moderate to severe hemolysis. The characteristic finding in hereditary elliptocytosis is elliptical or cigar-shaped RBCs on the peripheral blood film. The number of elliptocytes does not necessarily correlate with disease severity. Investigation of elliptocytosis involves a thorough patient and family history, physical examination, and peripheral blood film examination of the parents. Other laboratory tests may be necessary to rule out other conditions in which elliptocytes may be present. In the homozygous or compound heterozygous states, the anemia is moderate to severe, and excessive hemolysis is present. Patients with the HPP phenotype show extreme poikilocytosis, fragmentation, microspherocytosis, and elliptocytosis. RBCs in the HPP phenotype show marked thermal sensitivity, and RBC fragments are observed after incubation at 41 degrees Celsius to 45 degrees Celsius. Patients with moderate or severe hemolytic anemia due to hereditary elliptocytosis can develop cholelithiasis, hemolytic, aplastic, and megaloblastic crises. Hereditary ovulocytosis, Southeast Asian ovulocytosis. Hereditary ovulocytosis or Southeast Asian ovulocytosis, SAO, is a condition that makes the red blood cell membrane more rigid and resistant to malaria invasion, caused by a mutation in the gene for band 3. SAO is common in the malaria belt of Southeast Asia, with a prevalence that can reach 30%. It is inherited in an autosomal dominant pattern, and all identified patients are heterozygous. The SAO mutation causes an increase in membrane rigidity that may be due to tighter binding of band 3 to ankyrin or decreased lateral mobility of band 3 in the membrane. The exact mechanism is not fully understood. In patients with SAO, hemolysis is usually mild or absent. The characteristic oval-shaped red blood cells with 1 to 2 transverse bars or ridges usually make up about 30% of the RBCs, as seen on a peripheral blood film. No treatment is necessary for SAO. Mutations that alter membrane transport proteins. Overhydrated hereditary stomatocytosis, OHS. RBC volume is regulated by membrane proteins that control the intracellular concentration of cations, especially sodium. Mutations that affect these membrane transport proteins can cause premature hemolysis of RBCs. Hereditary stomatocytosis is a group of conditions where RBC membrane leaks monovalent cations, leading to overhydration or dehydration of RBCs. Overhydrated hereditary stomatocytosis, OHS, is an autosomal dominant condition where RBCs are excessively permeable to sodium and potassium, leading to overhydration and swelling. The exact molecular defect causing OHS is not known, but mutations in the RHAG gene have been found in some patients. Clinical and laboratory findings of OHS include moderate to severe hemolytic anemia, 5 to 50% stomatocytes on the peripheral blood film, macrocytes, decreased MCHC, reticulocytosis, and increased net cation content in erythrocytes. Splenectomy in OHS should be carefully considered due to an increased risk of thromboembolic complications. Dehydrated hereditary stomatocytosis, DHS, or hereditary xerocytosis, HX. A type of hemolytic anemia that occurs due to a defect in membrane cation permeability, resulting in dehydrated red blood cells, RBCs. DHS/HX is the most common form of stomatocytosis. The RBC membrane in DHS or HX is excessively permeable to potassium, causing water to be lost from the cell due to the reduced intracellular cation concentration. Mutations in the piezo-1 gene that codes for the piezo-type mechanosensitive ion channel component 1 protein in the RBC membrane cause DHS or HX. 2 mutations have been identified to date. Clinical features of DHS or HX include mild to moderate anemia, reticulocytosis, jaundice, and mild to moderate splenomegaly. Fetal loss, hydrops fetalis, and neonatal hepatitis may also occur. Laboratory findings in DHS or HX include elevated mean corpuscular hemoglobin concentration, MCHC, decreased osmotic fragility, stomatocytes, usually fewer than 10%, target cells, burst cells, desiccated cells with spicules, and RBCs in which the hemoglobin appears to be puddled in discrete areas on the cell periphery. Most patients with DHS or HX do not require treatment. Splenectomy is contraindicated as it does not improve the anemia and increases the risk of thromboembolic complications. Other hereditary membrane defects with stomatocytes. Familial pseudohyperkalemia. Excessive potassium leaks out of RBCs at room temperature in vitro but not at body temperature in vivo. CBC parameters are usually normal, with occasional stomatocytes seen on peripheral blood film. Gene defect associated with disorder not yet identified. Cryohydrocytosis. Mild to moderate hemolytic anemia with leakage of sodium and potassium from RBCs and stomatocytosis. RBCs show marked cell swelling and hemolysis when stored at 4 degrees Celsius for 24 to 48 hours. Mutations in band 3 or membrane stomatin deficiency have been identified in some patients. Rh deficiency syndrome. Group of rare hereditary conditions in which Rh membrane protein expression is absent or decreased. Amorph and regulatory types exist, caused by mutations in Rh proteins or a protein that regulates Rh gene expression, respectively. Mild to moderate hemolytic anemia observed, with stomatocytes and occasional spherocytes seen on peripheral blood film. Symptomatic patients may be treated by splenectomy, which improves the anemia. Acquired red blood cell membrane abnormalities. Acquired red blood cell membrane abnormalities include stomatocytosis, spur cell anemia, and paroxysmal nocturnal hemoglobinuria. Acquired stomatocytosis. Stomatocytosis is a common artifact seen on blood films stained with white stain. To accurately diagnose stomatocytosis, a medical professional must examine multiple areas on several blood films, as true stomatocytes should be present in all areas of the blood film. In normal individuals, 3% to 5% of RBCs may be stomatocytes. Wet preparations of RBCs in their own plasma, examined under phase microscopy, can help differentiate true stomatocytes from artifactual ones. Acute alcoholism and various medical conditions, including malignancies and cardiovascular disease, and medications have been linked with acquired stomatocytosis. Spur cell anemia. Severe liver disease can cause hemolytic anemia with acanthocytosis known as spur cell anemia in some patients. Acanthocytes are RBCs with a defect in the lipid component of the membrane due to excess free cholesterol in severe liver disease. The equilibrium between free cholesterol in plasma and RBC membrane cholesterol causes cholesterol to accumulate in the outer leaflet of the membrane. The spleen remodels the membrane into an acanthocyte shape, which results in hemolysis and a rapidly progressive anemia of moderate severity, splenomegaly, and jaundice. Spur cell anemia in end-stage liver disease has a poor prognosis, but the anemia may resolve if the patient undergoes liver transplantation. Paroxysmal nocturnal hemoglobinuria. Paroxysmal nocturnal hemoglobinuria, PNH, is a rare type of chronic intravascular hemolytic anemia. It is caused by an acquired clonal hematopoietic stem cell mutation which results in blood cells that lack glycosylphosphatidylinositol, GPI, anchored proteins such as CD55 and CD59. The absence of CD55 and CD59 on the surface of the red blood cells makes them susceptible to spontaneous lysis by complement. The mutation occurs in a hematopoietic stem cell, therefore the defect is also found in platelets, granulocytes, monocytes, and lymphocytes. PNH is uncommon and has an annual incidence of 2 to 5 new cases per million persons in the United States. Pathophysiology of the hemolytic anemia. Hemolytic anemia occurs due to a mutation in the GPI anchor, which consists of a phosphatidylinositol, PI, molecule and the glycan core. The glycan core contains glucosamine, 3-mannose residues, and ethanolamine phosphate. At least 24 genes are involved in the biosynthesis of the GPI anchor. PNH is caused by a mutation in the PIGA gene, which codes for phosphatidylinositol glycan class A PIGA, a subunit of a glycosyltransferase enzyme. The mutation results in a deficiency of membrane GPI anchors on hematopoietic cells. Without GPI anchors, hematopoietic cells are unable to express GPI-anchored proteins, which include complement regulators, enzymes, adhesion molecules, blood group antigens, or receptors. 2 GPI-anchored proteins, decay accelerating factor, DAF, or CD55, and membrane inhibitor of reactive lysis, MIRL, or CD59, are absent or deficient on the RBC membrane in PNH. CD55 inhibits the complement alternate pathway C3 and C5 convertases, and CD59 prevents the formation of the membrane attack complex. The absence of CD55 and CD59 on the RBC surface leads to the activation of complement and spontaneous and chronic intravascular hemolysis. PIGA is the only gene needed for GPI anchor synthesis that is located on the X chromosome, which means that only one acquired mutation in a stem cell is needed for the PNH phenotype. In PNH, there coexists a mutant clone and normal hematopoietic stem cells, resulting in a population of GPI-deficient RBCs and a population that is normal. The severity of chronic hemolytic anemia varies between patients depending on the percentage of circulating GPI-deficient RBCs. PNH patients display phenotypic mosaicism due to different PIGA mutations in mutant clones. This results in 3 RBC phenotypes: type 1, normal; type 2, partial deficiency of CD55 and CD59, and type 3, complete deficiency of GPI anchor. The severity of hemolysis depends on the relative amount and type of circulating RBCs. In addition to hemolysis, bone marrow dysfunction may contribute to the severity of anemia in PNH patients. Many patients have a history of bone marrow failure caused by acquired aplastic anemia or myelodysplastic syndrome that precedes or coincides with the onset of PNH. Clinical manifestations. PNH usually starts in the 3rd or 4th decade of life, but it can occur at any age. The main clinical manifestations of PNH are hemolytic anemia, thrombosis, and bone marrow failure. The severity of anemia depends on the type of RBC, the degree of hemolysis, and the presence of bone marrow failure. Free hemoglobin released during hemolytic episodes can remove nitric oxide and cause various symptoms like dysphagia, erectile dysfunction, and abdominal pain. Thrombosis is a common complication of PNH, and the most common type is hepatic vein thrombosis, which can be fatal. Laboratory findings. Laboratory findings in PNH include evidence of intravascular hemolysis such as hemoglobinuria, hemoglobinuria, decreased serum haptoglobin, and increased levels of indirect bilirubin and lactate dehydrogenase. Hemoglobinuria occurs in only 25% of patients at diagnosis but is present in most patients during the course of the illness. Hemosiderunuria may be detected with Prussian blue staining of the urine sediment due to chronic intravascular hemolysis. Reticulocyte counts are mildly to moderately increased, and the MCV may be slightly elevated due to the reticulocytosis. The direct antiglobulin test is negative. Confirmation of PNH requires the demonstration of GPI-deficient cells in the peripheral blood, determined by flow cytometric analysis of RBCs with fluorescence labeled anti-CD59. Type 1 cells with normal expression of CD59 show the highest intensity level of fluorescence, type 2 cells with a partial deficiency of CD59 show moderate fluorescence, and type 3 cells with no CD59 are negative for fluorescence. Patients with a greater proportion of type 3 cells are expected to have a high-grade hemolysis, while patients with a high percentage of type 2 cells and a low percentage of type 3 cells may have only modest hemolysis. The bone marrow aspirate and biopsy specimen are examined for evidence of an underlying bone marrow failure syndrome, abnormal cells, and cytogenetic abnormalities. An abnormal karyotype is found in 20% of patients with PNH. Flow cytometry methods to detect PNH RBCs have two major disadvantages. They underestimate the percentage of type 3 cells because RBCs lacking CD59 undergo rapid complement lysis in the circulation, and these methods cannot accurately determine the percentage of PNH RBCs after recent transfusion. Flow cytometry methods to detect PNH RBCs have inherent problems, so diagnosis of PNH is achieved using multiparameter flow cytometry to detect the absence of GPI-anchored proteins on WBCs. The absence of at least 2 GPI-anchored proteins in 2 WBC lineages, usually granulocytes and monocytes, is recommended for greater diagnostic accuracy. Fluorescent monoclonal antibodies to GPI-anchored proteins, CD59, CD55, CD24, CD16, CD66b, CD14, and lineage-specific antibodies to non-GPI-anchored proteins, CD15 and CD33 or CD64, are typically used to identify granulocytes and monocytes, respectively. An alternative flow cytometric method uses a fluorescein-labeled proaerolysin variant, FLAR, that binds directly to the glycan core of the GPI anchor with a high signal-to-noise ratio. The absence of binding on granulocytes and/or monocytes indicates GPI deficiency. Multiparameter flow cytometry with FLAER in combination with monoclonal antibodies to GPI-anchored antigens and lineage-specific antigens increases sensitivity and specificity for detection of GPI-deficient granulocytes and monocytes. A high-sensitivity four-color protocol using FLAER, CD24, CD15, and CD45 for granulocytes and FLAER, CD14, CD64, and CD45 for monocytes was able to detect GPI-deficient granulocytes and monocytes with a sensitivity of 0.01% and 0.04%, respectively. The sugar-water test and the HAM test have insufficient sensitivity for diagnosis of PNH and have been replaced by flow cytometric techniques. The results of the multiparameter flow cytometry with FLAER are not affected by recent transfusions and can be reliably used to estimate the percentage of GPI-deficient granulocytes and monocytes in recently transfused patients. The high sensitivity of the multiparameter flow cytometry with FLAER is also important for post-therapy monitoring of PNH clones. Thank you for completing the video. Remember to ask for ASCP short notes, and don't forget to subscribe. Bye.